For all things health and fitness, this is the Army Gymnastics Podcast.
The Army Gymnastics Podcast.
Discover a fitter, healthier you at armygymnastics.com.
Welcome to the Army Gymnastics Podcast. Today we're diving into something really fundamental, our brain health.
It really is. Everything starts there.
Yeah, think about it. Every move, every thought, it all hinges on this amazing organ. And, sometimes we might not realize just how much our lifestyle, things like exercise, can impact it.
Absolutely. We focus a lot on physical fitness, muscles, endurance, which is great.
Right.
But the brain is the command center. Keeping it healthy impacts, everything.
Which is why we're calling this deep dive Breaking Barriers, Brain Health, BBB, and Exercise Boost. We're going to explore some, frankly, surprising ways physical activity can protect our brains.
And a key part of that protection involves something called the blood-brain barrier, or BBB.
Right, the BBB. Sounds technical, but maybe think of it like super tight security for your brain.
That's a good way to put it. It's a layer of specialized cells lining the tiny blood vessels in the brain, acting like a very selective border control.
Controlling what gets in, what stays out, crucial stuff.
Exactly. It keeps the brain's environment stable and protected. And what's really exciting, and what we'll get into, is how exercise seems to positively influence this barrier.
Leading to real brain benefits. Okay, let's unpack that. Starting with the BBB itself. How did we even figure out it existed?
Well, the initial understanding came from way back, classic experiments. Scientists injected dye into the bloodstream and noticed the whole body stained except the brain. Then, if they put the dye directly into the brain fluid...
Then the brain stained, but not the body.
Precisely. So that strongly suggested some kind of barrier.
So the first thought was just like a physical wall, impenetrable.
That was the early concept, yeah. A simple physical block. But we know now it's much more sophisticated. It's not passive at all.
Okay, so what does it do then, besides just blocking?
It actively manages traffic. It has these tiny transport systems built into the vessel cells. Think specialized doorkeepers.
Doorkeepers, I like that.
Yeah, moving ions, charged particles. And then there are these things called ABC transporters. They're like little pumps that actively push certain unwanted molecules out of the brain.
So it's defense, but also active regulation.
Exactly. Plus, the cells themselves have enzymes, like tiny processing plants that can break down molecules trying to cross. Metabolic activity right at the barrier.
Wow. Like customs officials who can dismantle suspicious packages on the spot?
Yeah, that's a good analogy. And it gets even more complex. These cells have different faces, one side toward the blood, the other towards the brain.
Luminal and abluminal, right?
That's the term. And they have different sets of transporters on each side. This allows for really coordinated movement, like how glucose, the brain's fuel, gets in via the GLUT1 transporter, very specifically managed.
Okay, that's pretty intricate for small molecules. What about bigger things like proteins or peptides? How do they ever get across?
Right, that's where vesicular transport comes in. Tiny bubbles, basically. They form on one side, engulf a molecule, travel across the cell, and release it on the other side.
Like a little ferry service?
Sort of. There are two main ways: receptor-mediated transcytosis, RMT, and adsorptive-mediated transcytosis, AMT. RMT is like having a specific key. A molecule binds to a receptor, and that triggers the uptake.
So it needs the right ticket to get the ferry.
Exactly. AMT is more about molecules, often positively charged ones, just sticking to the cell surface and getting pulled in.
Okay.
And this RMT process, the one needing the key, that's where the idea of Trojan horses comes in for drug delivery.
Trojan horses? Sounds sneaky.
It kind of is. Scientists try to design molecules, maybe engineered proteins or antibodies, that can grab onto those RMT receptors, then they attach a drug to it.
So the barrier lets the horse in, but it's carrying the medicine.
That's the goal. Disguise the drug as something that BBB naturally transports. They're targeting things like insulin receptors or transferrin receptors.
Clever. Does it work well?
It's promising, but tricky. These systems can be species-specific, meaning what works in the mouse might not work the same way in us. It's still a big area of research.
Makes sense. So with all this complexity, how do we know if the barrier is actually, working properly? How do you test its integrity?
There are a few ways. We can use small marker molecules like sodium fluorescein. It's small, doesn't stick to much, so if it leaks into the brain, that tells you something's wrong.
Like a tracer dye?
Yeah. Or labeled dextrans, which are sugars you can track. If they show up where they shouldn't, it's a sign of leakiness.
Okay. What about sampling fluids?
Right, you can look at the cerebrospinal fluid, the CSF, usually via a lumbar puncture. Certain proteins shouldn't really be there in high amounts if the BBB is tight. S100 is one example.
That's 100 beta.
Yeah, it's normally kept out. If levels are high in the blood, it suggests a leaky BBB. There's another one, NSE, which is inside neurons.
Right.
So if the BBB leaks but the neurons aren't damaged, you might see S100 go up, but NSE might stay relatively normal. Helps differentiate.
Ah, that's useful. And imaging, brain scans.
Yep, contrast-enhanced CT or MRI. You inject a contrast agent, and if the BBB is broken down, the agent leaks into the brain tissue and shows up on the scan.
Okay, so we have this incredible dynamic shield, but obviously things can go wrong. What kinds of things lead to BBB impairment?
A lot of neurological conditions involve BBB problems. Take brain tumors. The tumor cells themselves can release substances like VEGF or cytokines.
Signaling molecules?
Exactly. And these can actually disrupt the tight connections between the blood vessel cells. Makes the barrier leaky.
So the tumor actively sabotages its own environment's defenses.
Seems that way. Plus a water channel protein, aquaporin-4, seems involved in the swelling, the edema around tumors.
And it's not just tumors, right? You mentioned other conditions?
Oh, yeah. Multiple sclerosis, MS, involves issues with those tight junctions, loss of structural support proteins.
Yeah.
In HIV, inflammation triggered by the virus can damage the barrier.
Inflammation again. Seems like a common culprit.
It really is. And in Alzheimer's disease, we see problems too, like decreased transport of glucose across the BBB because the GLUT1 transporter levels go down. Other proteins involved in barrier function are also altered. It's complex, varies by disease.
And this impairment explains why some drugs don't work for brain conditions, even if they work elsewhere.
Absolutely. Many chemotherapy drugs, for example, that are effective systemically just can't cross an intact or even partially intact BBB in high enough concentrations. Trastuzumab for certain breast cancers is a classic example. Even when cancer spreads to the brain, the BBB within those metastases can still be a major hurdle.
So the BBB is this double-edged sword, vital protection, but also a barrier to treatment. Okay, let's shift gears to something really intriguing, this runner plasma idea. What's that about?
This is super fascinating. Researchers took plasma, the liquid part of blood from mice that were voluntarily running,
Okay, active mice.
and infused it into sedentary mice, mice that didn't exercise. And remarkably, the sedentary mice started showing some of the brain benefits you normally only see with exercise itself.
Whoa, wait, so something in the blood of the runners was helping the brains of the non-runners.
That's exactly what the studies suggest. This runner plasma reduced baseline neuroinflammation, the sort of background inflammatory state in the brain.
Okay.
And it also suppressed inflammation that was deliberately induced in experiments.
So it's like exercise creates this transferable anti-inflammatory cocktail in the blood.
That seems to be the case. When they analyzed the proteins in the runner plasma, they found a big increase in inhibitors of the complement cascade.
The complement cascade sounds dramatic.
It's part of our immune system, really important for fighting pathogens. But if it gets overactive, it can drive inflammation. So inhibitors keep it in check.
And one specific inhibitor stood out.
Yes, a protein called clusterin. It was significantly higher in the runner plasma, and further tests showed it was actually necessary for those anti-inflammatory effects.
Clusterin. Okay, remember that name. Does this apply to humans too? Or just mice?
Well, there's evidence in humans too. A study looked at people with mild cognitive impairment who did a 6-month structured exercise program.
Right.
Their plasma clusterin levels increased after the exercise. And interestingly, the increase in clusterin correlated positively with improvements in their endurance, how far they could walk.
That's a strong link. Suggests these exercise factors are real in humans and tied to functional benefits.
It really does. It points towards transferable anti-inflammatory factors generated by exercise in us, too.
And it's not just clusterin, right? You mentioned other proteins increase too.
Yes, dozens of proteins change in the plasma after exercise. Many are likely myokine signals released from working muscle, things like IGF-1, VEGF, PF4. These have been linked to increased neurogenesis, making new brain cells, especially in the hippocampus.
The hippocampus key for learning and memory. So exercise signals the muscles. Muscles release factors into the blood. Blood travels to the brain. And factors like clusterin reduce inflammation, while others potentially boost new cell growth.
That's a great summary of the working hypothesis. It's a cascade of benefits.
Okay, let's dig into how that runner plasma worked in those mice studies. What specific changes did they see in the brain?
They saw more cell proliferation, new cells being born in the hippocampus of mice, getting runner plasma compared to control plasma. specifically EDU-positive cells.
EDU is a marker for new cells.
Yeah. They also saw more neuroblasts marked by DCX, those are young neurons, and better survival of cells born earlier, marked with BRDU. Very similar effects to what happens when mice actually run.
So the plasma really recapitulated key effects of running. Was there a time element? Like, did it matter how long the donor mice ran?
Interesting point. They found that plasma from mice running only 7 or 14 days mostly increased the neuroblasts, but didn't boost overall proliferation and survival as much as plasma from longer-term runners. Suggests the profile of factors might evolve.
Okay. And what about the fate of these new cells?
The runner plasma led to about a twofold increase in the survival of neural stem and progenitor cells, the brain's reserve pool, and it helped them mature into neurons. It also boosted the survival of new astrocytes, those important support cells.
So promoting not just birth, but survival and maturation, did it affect actual thinking, cognition?
Yes. They used a fear conditioning test, which assesses learning and memory related to context. Mice getting runner plasma showed significantly better memory for the context compared to controls.
Wow. Consistent with exercise-boosting learning and the hippocampus link.
Exactly. And it seems specific it didn't affect their response to simple cues or their general activity or anxiety levels.
That specificity is important. What about genes? Did they look at what genes were turned on or off in the hippocampus?
They did, using RNA sequencing, found nearly 2,000 genes with different activity levels in the runner plasma group, and interestingly, most of them were downregulated, less active.
Less active, huh? What kinds of genes?
Things involved in cell migration, cell adhesion, some types of cell differentiation were downregulated, but genes related to synapse function, like FOXO6, and immune regulation or plasticity, like FAM19A2, were upregulated.
A mix, then. What was the overall pattern?
Looking at the broader functions, the gene ontology analysis, there was a significant downregulation of the acute inflammatory response. That really stood out. And an upregulation of things like organizing cell junctions and controlling protein breakdown.
So calming inflammation and promoting stability at the genetic level.
Okay, you mentioned inflammation. Did they test if runner plasma could reverse existing inflammation?
Yes, that was a key experiment. They used LPS, a bacterial component, to induce inflammation systemically, which also affects the brain.
Standard model for inflammation research.
Right. LPS caused widespread changes in gene expression in the hippocampus, as expected.
Yep.
Then they gave runner plasma, or control plasma.
The results were pretty striking. Runner plasma, but not control plasma, reversed the expression of many of those LPS-induced genes. The gene profile in the LPS plus runner plasma mice looks much more like the profile of mice that never got LPS at all.
That's a powerful effect, like hitting a reset button on the inflammation. Any specific pathways?
They confirmed, using qPCR, runner plasma reduced activity in the toll-like receptor pathway, think MyD88, TLR1, TLR2, also dampened the interferon pathway, ERF9, STAT1, STAT3, and lowered GFAP, the astrocyte activation marker.
So hitting multiple key inflammatory routes.
Exactly. The overall pathway analysis suggested a broad dampening of the response to cytokines and better regulation of the innate immune response.
Which brings us back to clusterin. You said it was needed for the anti-inflammatory effect.
Precisely. The detailed protein analysis, the proteomics, identified 49 proteins significantly changed in runner plasma. Lots involved complement and coagulation.
Complement again. Seems central.
It does. Pathway analysis pointed to activation of the alternative complement path and endothelial cell changes. So they did the crucial experiment. Removed clusterin from the runner plasma.
Took out the key suspect.
And when they did that, the ability of the runner plasma to suppress those LPS-induced inflammatory genes, TLR2, STAT1, GFAP, was largely abolished. Gone.
Wow. That really pins clusterin as a major player in mediating exercise's anti-inflammatory brain benefits, at least in this model.
It's strongly suggested, yes. And tying back to the human study.
With the cognitively impaired patient.
Yes. Remember, their clusterin went up with exercise, and that correlated with better endurance.
Right.
They also found that the change in clusterin in those individuals correlated with changes in other immune-related proteins in their blood, suggesting as part of a coordinated systemic response to exercise.
So exercise shifts this whole network of immune and complement factors, and clusterin seems to be a key hub, particularly for the anti-inflammatory effects impacting the brain.
That's a good way to visualize it. There are likely other factors involved too, but clusterin emerges as very important.
This is incredibly hopeful. It suggests mechanisms we might be able to target. Now, slightly shifting, but related to brain health and biomarkers, there was some buzz, I think, on Reddit about predicting dementia years in advance using proteins. Does that connect here?
It does connect in the broader sense of using blood biomarkers for brain health. That discussion highlighted studies looking for patterns of proteins in blood that might predict dementia risk, maybe even 15 years out.
Wow, 15 years. That's significant.
It is. And someone shared a personal story about their mother developing vascular dementia, emphasizing the human impact. Vascular dementia, involves problems with blood flow in the brain, often from small strokes.
Which links back to the health of brain blood vessels, potentially the BBB itself.
Exactly. The hope is that if we can identify risk that early, maybe interventions, lifestyle changes, perhaps future drugs could slow things down. Though current dementia meds have limitations, maybe early intervention would be key.
Yeah, the discussion probably touched on lifestyle too. Sleep, diet.
Definitely. Sleep's role in brain cleaning came up. And ethical concerns too, like could insurance companies use this risk information? Plus the financial burden of long-term care.
All valid points. Did exercise come up in that dementia prediction context?
Yes, someone mentioned cardio exercise and neurogenesis, tying back nicely to what we've been discussing. Also, interesting links were mentioned between hearing loss and dementia risk, suggesting interventions like hearing aids could matter, and some studies linking long-term use of certain meds, like blood pressure or cholesterol drugs, to reduce dementia risk.
So it's a complex picture: genetics, lifestyle, vascular health, maybe even sensory input like hearing. But the idea of biomarkers, like perhaps clusterin or others, being indicators is powerful.
Okay, let's zoom back to the BBB itself in the context of neurodegenerative diseases more broadly.
Right, so we've established the BBB isn't just a wall. It's a physical barrier, yes, but also metabolic with selective transport. And it acts as a signaling interface between the blood and the brain. And it works with other cells, right, the neurovascular unit.
Exactly. The BBB endothelial cells, neurons, microglia, astrocytes, they all work together. The gliovascular unit focuses on the astrocyte-BBB interaction. They regulate blood flow, maintain the environment.
So a key question in diseases like Alzheimer's or Parkinson's is, does this unit break down? Does the BBB function change, maybe differently in different brain regions?
Those are the big questions. We know neuroinflammation is a feature of these diseases, with more immune cells getting into the brain. That implies the BBB is compromised somehow.
Allowing things in that shouldn't be there.
Yes, and maybe allowing things out too. There's a concept that brain-specific antigens might leak out and trigger immune responses elsewhere in the body, potentially contributing to the disease process, like in Parkinson's.
And what about factors that regulate blood vessel growth? Angiogenesis.
Yeah, there might be an imbalance between factors that promote vessel growth, like VEGF, and those that inhibit it. This could lead to chronic BBB dysfunction and leaky abnormal vessels worsening the degeneration. VEGF might be a player there too.
Okay, let's focus specifically on Alzheimer's disease now. There's a strong link emerging between vascular problems and the amyloid beta pathology, right?
A very strong link. We now know amyloid beta itself, the protein that forms plaques in AD, can directly affect blood vessels. It can cause vasoconstriction, narrowing of the vessels.
Wow.
It seems to increase damaging free radicals and boost the action of endothelin-1, a potent vasoconstrictor. It also activates inflammatory pathways involving enzymes like COX2.
So the plaque protein itself messes with blood flow regulation.
Yes, and it can also affect the smooth muscle cells around larger vessels and the pericytes around capillaries, making them contract more or respond poorly. This could contribute to the reduced overall cerebral blood flow we often see early in AD.
And the BBB's role in clearing amyloid beta is also affected.
Critically so. Normally, the BBB helps transport aft out of the brain, primarily via a receptor called LRP1 on the endothelial cells.
LRP1.
In AD, LRP1 levels are often downregulated, reduced. AP itself seems to promote the breakdown of LRP1. So less LRP1 means less clearance, leading to more AP buildup in the brain.
A vicious cycle. The thing that needs clearing damages the clearance mechanism.
Exactly. There's another player called pineolm involved in this LRP1 transport process. Pineolm levels are also reduced in AD brains. Interestingly, there's a protective genetic variant of pineolm that seems to boost AP clearance.
Fascinating. So genetics can influence this clearance pathway.
Yes. And other factors too. Low expression of a gene called AOX2 involved in vessel development can also lead to less LRP1 and more AP retention. The bottom line is impaired AP clearance at the BBB is a major contributor to Alzheimer's pathology.
And this is on top of other vascular issues in AD, like microdamage, inflammation.
Loss of pericytes, those capillary regulating cells. And remember, the reduced glucose transport we mentioned earlier. Lower GLUT1 levels mean less energy for brain cells, which likely worsens both neuronal and vascular dysfunction. It's all interconnected.
It really paints a picture of the brain's vascular system, including the BBB, being deeply involved in AD, not just a bystander. Okay, this has been incredibly insightful.
It really highlights how crucial vascular health is for brain health.
So let's try to wrap this up. The key takeaway seems clear. Exercise has this profound positive impact on brain health. And it works, at least partly, by influencing factors in our blood, like cholesterol.
Yes, these factors can dampen inflammation and potentially protect the integrity of that vital blood-brain barrier.
Which is just amazing. The idea that transferable factors could mimic exercise benefits opens up really exciting possibilities for future therapies.
Absolutely. It's a very active area of research.
So for everyone listening, the message really is about prioritizing physical activity. It's not just for your body, it's a serious investment in your long-term brain health and resilience against these complex processes.
Making that connection is so important.
It really makes you think, doesn't it? What's one small step you can take today? Maybe a walk, maybe trying something new. Just move more for a healthier brain tomorrow.
Every little bit likely helps build that resilience.
For expert fitness advice and personalized plans, visit armygymnastics.com, your go-to resource to reach your goals. Start your journey today at armygymnastics.com.
Thanks for listening. For all things health and fitness, subscribe to our newsletter today. Find out more at armygymnastics.com.